Most people will know that you have a genome code that is present within every single cell, and it often is, well, often is always the same in every single cell. But cells are very different and they behave differently, and that's because of the epigenomes. In other words, how exactly that code is read. So the epigenetic regulation of things is driven in large part by methylation as one of the mechanisms. So it really tightens up the DNA, makes it more difficult for it to be transcribed and read and the code to be passed along to the cell. And so we looked specifically at DNA methylation to work out whether or not we can have a good understanding of which genes are switched on and which genes are not. And to do that, we did a methylation screen, but then we also partnered that with the transcriptome. So in other words, we looked to see where we thought the methylation was happening for the promoter regions of the various genes, and then we looked specifically to say which genes seemed to be transcribed, and we actually had evidence that that gene was switched on or switched off. And in essence, what we found when we compared normals to low-grade IgM MGUS to Waldenström's patients, that as you go to the MGUS phase, there is an increased amount of cytokine production and pathways that really suggest inflammation and activation. And then as you get to the true Waldenström stage, a lot of that switches back off again. So it really fits very nicely with some of the conversations that have been happening around the memory B cell type of Waldenström's and a plasma cell type of Waldenström's, where we see that this transition toward memory B cells and then on toward plasma cells may be inflammatory mediated and mediated by a lot of the cytokines that are in turn regulated by DNA methylation.